In this video, we're going to be reviewing the function of histones in gene regulation. Now, we're going to start with the idea that chromosomes are typically not found in the cell that look like chromosomes. So normally we kind of think of a chromosome having this kind of shape like this. Here it is on its side, and here we have kind of unwound it so that we see that the DNA is wrapped around the histones. The histones, there are usually 8 of these little proteins stuck together. They have it shown here as a little ball, but realize these are actually multiple proteins making up the histones, the histone proteins. And when you wrap the DNA around it, you call it a nucleosome. But the normal structure of the DNA is not in the form like this. Usually the chromosomes are actually strung apart and they're more loosely wound than you see it right here. They can be all over the nucleus like a phone cord that is, or an extension cord that has been, unwound and it's kind of laying in the floor. That's the normal state of a chromosome. It is not nicely wound up tight like you would be when you were like winding it up to carry it or store it. That would be the chromosome that we kind of look like here. This is the wound up part, and that is not the normal case. Normally it's unwound and it's being used. When it's wound up, it's not being used. It's actually, if you see the chromosome like this down here, it's undergoing replication. In other words, you're copying it, and that is not the normal state. Normally when you're like transcribing genes into RNA and translating that RNA into proteins, the DNA is going to be unwound, at least parts of it are. Some of it may be compacted like this and may remain that way, but the majority of it's going to be unwound, at least to some extent. And depending on how much, of course, that you unwind it, the more access various proteins in the nucleus have access to the genes. In other words, when it's wound up really tightly, especially when it's being replicated, proteins don't have access to the genes, to the DNA, and in that case RNA polymerase wouldn't, and if RNA polymerase doesn't, those genes are not on, of course. And so depending on how tightly you wrap the DNA around these histone proteins and form nucleosomes, depends on how well transcription occurs. And if you don't do transcription well, you're not going to be making many proteins. Each gene transcribes into an RNA, and then that RNA is translated by a ribosome into a protein. And if the RNA polymerase, which is the primary protein that does transcription, if it cannot get access to the DNA in here, it is not going to create, that gene is not going to be on. That gene will be off. Now, of course, if you're the DNA in between the histones, if there's a gene in this region, all right, well, that would be on. It's obviously not wrapped around a histone at all. Now, you can control how well different proteins have access to the DNA by how tightly you wrap the DNA around the histones. And that's what we're going to look at here. We're going to look at the things that cause the DNA to wrap tightly or loosely around the histones. So keep in mind, open or free DNA, or we call it chromatin, open chromatin, is free for RNA polymerase to come in here and attach to the DNA. So this would be free or open chromatin. Here we have the DNA wrapped around that, and if that's wrapped very tightly, we would call that maybe closed chromatin. And so we want to find places that are open and closed when we're trying to understand how which genes are turned on and off in the chromosome, in the genome of a particular cell type. So different cells, of course, have different DNA turned on and off, and what we're really looking at is how do those cells that have certain genes turned off so that they can be a skin cell or a a liver cell. How do they do that? How are the histones related to this? So let's go to the next slide. Here is just the chromatin structure. Let's zoom in just a little. And alright, and I'll go back. Now here it's, notice it's giving us some names, H2A, H2B, etc. These are the types of histones. There are 4 histones, 2 H2As, 2 H2Bs, an H3 and an H4. There's also an H1, which usually is on the outside of the DNA. But this right here is the H1 right here, if you can see that. Alright, so these others are on the inside, and there are different versions of these H2A. So there might be an H2A 1B or something. They'll give them different names, H4C or something. But the idea is they're all a little different, but you need 8 in there to form a nucleosome, which is the histone proteins with the DNA. There's a certain distance here that we know of that's 10 nanometers, and we know octameric, of course, means 8 histone core. That includes the little external one, the H1. The H1 is very important at kind of locking this down. You might think it wraps around it and then you kind of like twist tie it or lock it here with the H1. But this is the structure. Now this DNA is more accessible to RNA polymerase and other proteins than this DNA. Notice how tightly these nucleosomes are all bundled together. We're really shutting down access to the other proteins when you cause the actual nucleosomes to bind to one another. So this, it would be more compacted than this, and that would be the way you would say this. This is very compacted DNA. This is less. This is more likely to be having to have ongoing transcription going on. In other words, the genes that are in the DNA over here are more likely to be active and expressed in the cell than the ones over here. So let's go on to the next slide here. This concept of histones is called the beads on a string. Alright, so beads on a string, it's kind of the idea that you have these beads and there's string wrapped around them, and you can compact the DNA when you create nucleosomes here. And when you create the nucleosomes, you can compact the actual nucleosomes together real tightly or leave them unwound. And where you actually wrap the DNA around it, you actually can control what is exposed. Now you'll notice here's a very large strand of DNA that is exposed, so that's going to probably be on. And you'll notice they have it where there are proteins binding. Now often these proteins are transcription factors and RNA polymerase and things like that, and so they call them sequence-specific DNA binding proteins. So that can be anything that would bind to the DNA, like a transcription factor, enhancer regions, proteins that bind to enhancers. And enhancers are very similar to promoters in that specific proteins bind there, but there are usually specific proteins binding in enhancers that will control an entire region. So the enhancer protein would be controlling what's going on in a much larger group. And what it's actually doing is that it's folding up, it's causing these histones to fold up or bend in a way so that they are tighter. We may get into some of the functions of enhancers, but enhancers are very much like transcription factors in that things bind to those regions and it causes the genes to either increase transcription or decrease transcription depending on what's going on in an enhancer, as it is in a promoter. A promoter, different things bind, transcription factors bind, and they either like increase or decrease transcription of that region. Alright, so let's move on. Alright, so let's zoom in a little more here. Let's get a little larger. Okay, well, this is just showing you that it's very compact here and it's uncompact here. It's looser, more loosely wrapped, And there are proteins that cause it to compact. Here, we have names for them. Compacting proteins, all different terms, but one of them that unpacks it, we should know, is hats. So we're just kind of introducing that hats would be anything that's kind of given that name is something that's going to cause the nucleosomes to kind of come apart and be a little bit looser in their binding, and therefore give us access to the RNA polymerase and other proteins. And as you can say, open chromatin, the genes are active. Closed chromatin, the genes are silent. So keep that in mind. Here we go. Hats, of course. If we have hats binding to things, like here we have a transcription factor, and then a hat will bind to it, which will essentially activate this region. All right. Now over here we have it compacted. The thing that causes the compaction, we call them HDACs or HDACs, however you want to say it. And again, it will bind to a transcription factor that will cause the deacylation. So notice we call the deacylation, and we call this acylations over here. Now acylation will alter the nature of the histones. So something that alters the tails on those histones, okay, we're going to talk about tails in a second, but if we're altering the acylation, we're altering the, if we're acylating the tail, all right, we're adding an acetyl group to probably a lysine, and when it's an amino acid that will be part of that tail on one of the histone proteins. When we acylate it, we're adding an acetyl group to it, and that's changing the chemical nature of it, and it's altering how well the DNA wraps around it, whether or not it wraps tightly or loosely, more or less. So we need to go on and kind of look at this. So here we're looking up, and you can see here's a tail, and we're showing that there is a lysine amino acid in that tail. So you have all these dots that are amino acids, and here you have one on here that's a lysine, and you will use a protein, an enzyme that would go and alter that lysine and add an acetyl group to it. So when you add acetyl groups, all right, it's going to alter it, and you'll notice here that it is more loosely compacted when the acetyl groups are not there. It is more tightly compacted, and therefore you have repressed gene transcription. In other words, lower transcription rates. Up here, you would have higher transcription rates. So HAT, HDAC, okay, kind of get the idea. All right, and as you look at it here, again, you can definitely see with this one that there's much more room here for transcription to go on. You've got fewer histones. Notice how up here I have lots of histones, and they're very compacted in the same area. Here I've got fewer. It's almost as if you took them out, which is exactly what happens at times. And you've added acetyl groups that have altered how tight, you might say, that everything is wound up. Realize there are lots of proteins involved in this. There's not just, we're not going into much detail here, but there are a ton of proteins involved in this. And as you can see here, you have your HDAC and your HAT, but you have transcription factors, and they're giving you very specific types of proteins here that we won't learn, but this is called chromatin remodeling, all right, and what you're doing is you're kind of changing the winding nature of it. You might also be changing the proteins that are involved in these histones, and you might be slightly adjusting the DNA that's wrapped around it versus what's loose. So there are lots of things that can be going on here, And the hats are essentially what we call writers, and the H-stacks are essentially called erasers. So you're either adding or removing acetyl groups to the lysines. That's one way to do this. There are several ways. So you also have things that are repressors, but the idea is there are the lysines with the acetyl groups. All right, that's going to kind of make it more easily read. If you take them off, they're not so easily read. All right, now you come over here on some more terms, Euchromatin versus Heterochromatin. It's just these terms come along with how much protein is actually bound to the DNA. If there's a lot of it, you're heterochromatin. If there's not so much, it's euchromatin. It's the same idea. And euchromatin is where you have transcription on. Now we introduce here the idea of Cpg Islands. And remember, it's not just the acetyl groups or the tails and altering them that affects transcription. You can alter the DNA as well. The DNA can be altered by methylating C's when the C's are next to a G. So when you have a C followed by a G, that C can be methylated by certain proteins in the nucleus, and those proteins come along and methylate it, and that would also alter how tightly, you might say, that the DNA wraps around these histones. So when you demethylate it, all right, so here we have demethylated CpGs, and we are in euchromatin, which would probably mean that we have acetylated lysines on our tails of the histones. We are more than likely in a region where transcription is on. If either you deacylate the histones or put the certain histones in here, depending on what proteins are located in the nucleosome here, and change the Cpg methylation pattern. Basically, you are going to, say, turn it off if you alter it in a particular way. Change the proteins, change the acetylation, change the methylation on the DNA, do those things, and you might turn it off. In other words, those are the things, the proteins present, the acylation of the tails, and the methylation of the Cpg Islands. Those are the things, depending on what you've done, you have either moved more towards closing off the DNA from transcription or opened it here. It's open up here, all right, but closed down in this region, and it's because of these modifications we've made. All right, a little bit more here. Let's zoom in a bit. Now, there's a lot going on here. We're looking at a single nucleosome, a chromatin unit, as it's called here. Notice the H1 is actually not in that group. It's out here, and it's called the linker, or at least the linker DNA is what's attached to the H1. And you can notice how that can either link it here or kind of close it off or not. And you see how it's unattached here, and it's more likely to be open chromatin or active chromatin when it's in this form. All right, so you have all kinds of things. You have different types of marks here, and this is how we look at those. So H4 represents the particular histone protein that we're talking about. Acylation on lysine 5. All right, there would be several lysines on those tails, and this is like lysine 5. Here we're talking about lysine 9, but we haven't acylated it, we've methylated it, and this is on H3. So it's a different protein, a different histone protein than here. Here, this one is also H3, and we've acylated the lysine at 9 and 14, I guess. So this is telling us that these, something about these nucleosomes and exactly what the nature of the of the nucleosome is in this area, and that of course is going to control what can get in and out and what can bind to it. And so this is a very complex process, as you can imagine, and we're just learning now what these marks, we're calling these histone marks. They're epigenetic marks. You can think of it as punctuation, and the punctuation, like say start a new paragraph, or put a period in, or put a comma or a semicolon, or start a new page even. All of that is kind of what we're talking about when we're talking about marks. So these are epigenetic marks. It's kind of like punctuation in something you're writing. Epigenetic marks tell you how to read your genes, whereas in grammar, grammatical marks tell you how to read your sentence, where to put a pause, where to put where your sentence ends, that sort of thing. And that's what's going on with these marks. That's the way we look at it when we talk about epigenetics. All right. Now here, do a little bit more, and I want to zoom in. Let me see here. Just not letting me zoom this one. Maybe I can stretch it. Nope, I can move it, but I can't stretch it. All right, so the idea with this is DNA methylation at the promoter, all right, is one of the marks that's important, but also here we have trimethylation, all right, at a particular histone. So trimethylation, you can imagine, it's 3 methyl groups added to a particular, say, lysine. In this particular case, here you have acylation and at this particular histone, and at this particular lysine 9, and here you have acylation at lysine 27. So you need to be able to read these and understand that. We really aren't going to go in that much detail, but you can see that these marks pretty much make it open chromatin, and here these marks compacted it quite well and closed off the chromatin, and that's what we're trying to see here. This is transcriptionally repressed, this is transcriptionally active. We call this open and this closed. So in a particular cell that doesn't need the genes in this region, you would close it off. If you're in a type of cell that needs these genes turned on, you would have marks very similar to this, which would open the chromatin. And here's kind of a review of all the things that we looked at. Let's see if we can get this a little larger without, let's see. All right. The idea here is there are writers and erasers, so there are proteins out there that will alter these marks and these things, and there were proteins out there that can alter the methylation of the DNA as well. And when they do these things, either alter the tails or alter the DNA, they're going to alter access to the polymerase and to the transcription factor proteins that want to bind in those areas. And when you restrict those things, then you are essentially turning that region of the DNA off. You're turning those genes off. When you open them up so that those proteins can get in there to contact the DNA, you have turned them on. And so these readers and these writers and these erasers are the things that basically turn the DNA off. Now we have nucleosome and histone modifications. These are called epigenetic marks on the histone. We also have methylation of the Cpg Islands. Realize there are other sequences besides C followed by G that can be methylated that can also do this, but by far in humans and in mammals, it's the methylation of C's and G's. And we do have other places that can be methylated that can alter it, but by and far it's C followed by G that does this. So between methylating the DNA and then marking up the histone tails, The other thing that could be part of that we have not talked about are non-coding RNAs, and we will talk about that in a different video. But realize compacting the DNA would also restrict contact with these RNAs. These RNAs are going to interact with the genes as well. We'll talk about that. But if you compact it, they also cannot interact. So these three things are all essentially need to have open chromatin to work, and if you close off the chromatin, basically you have closed how proteins and RNAs can get into there to interact. The non-coding RNAs work a little bit different, and we're just now learning how they work. We don't know a lot, but we are getting a pretty good grasp on how this works. And in fact, we understand it well enough that we can actually talk some cells into becoming stem cells and then talk them into being a different type of cell after that. So we are learning to very slowly re-engineer the cell. We take a cell, and it might be a skin cell, we turn it into a stem cell, and then after we turn it into a stem cell, we turn on a different set of genes and turn it into a liver cell or something. So that's the kind of thing that we learn with these readers and these writers, and we learn to alter the epigenetic marks here and in the DNA. Not so much in the RNA yet, but we're learning. But the whole idea is how it winds around the histones is essentially the primary method for turning genes on and off. And that's the big lesson here. And in all the details are not as important, just knowing that big picture idea that altering the histones, perhaps the DNA, are going to control how you wrap the DNA around the histone and how well you open it or close it up to other proteins and other things in which control transcription rates, or basically shut them down completely if you wrap it up very tightly. All right, and that's it for this video.